Welcome back, folks. We're going to wrap up this discussion of the cytoskeleton with a real quick look at intermediate filaments. We're not going to get into this in too much depth. Now, unlike microtubules and microfilaments, intermediate filaments differ greatly in their amino acid composition, and they vary hugely from tissue to tissue. We can basically group intermediate filaments into 6 classes, 5 of which are shown in the table here. Class 1 are acidic keratins, and Class 2 are the basic or neutral keratins. And proteins in Class 1 and Class 2 of the intermediate filaments make up the keratins that are found in the epithelial surfaces that cover the body and which line some of its cavities. You'll be intuitively familiar with the keratins in terms of skin structure. Class 3 includes proteins like vimentin, which is found in connective tissue, desmin, which is found in muscle tissue, glial, fibrillary, acidic protein, GFAP, also found in, obviously found in glial cells, sorry. Class 4, the neurofilament proteins, are found in nerve cells. Class 5 is the lamins, including nuclear lamins A, B, and C, and they form a network along the inner surface of the nuclear envelope, and so they line the inner surface of that structure. Now, you can distinguish animal cells based on the types of intermediate filaments that you find in the cells of the tissue, and that's called intermediate filament typing. So that just gives you a quick overview of the different types of intermediate filament. You don't necessarily need to memorize the classes and where you find them, but I would like you to really at least appreciate the acidic keratins and what they do, and then the nuclear lamins down here in class 5. So let's just take a look at a generic structure of an intermediate filament. So the basic structural unit of the intermediate filament is 2 intermediate filament polypeptides, and there's one individual one shown here, so it's shown in 2 diagrams. One is kind of like the schematic here, and then one here is showing some helices. So the basic structural unit of intermediate filament is 2 of these monomers associated and kind of twisted together to form this structural unit here. Then the dimer associates laterally with another dimer to form a tetramer. So there are 4 polypeptides here. And then we arrange the tetramer kind of end to end so that these kind of missing sequences or segments here overlap. So they form this kind of overlapping structure of 8 protein subunits, and that becomes called a protofilament. And then you can assemble the protofilaments into the actual intermediate filament by bundling together 8 protofilaments. So you can see this is an incredibly kind of beautiful example of quaternary structure in proteins. But even though there is no tertiary structure, because all we have here is alpha helix, so there's no folding of the alpha helices or beta sheets, because beta sheets are absent here. So here we have primary sequence, secondary sequence, no tertiary sequence, but in the mature form we do have quaternary sequence. So given the structure that you see here, hopefully you can appreciate how intermediate filaments probably play a significant role in generating tension, generating the ability to bear tension, so the tension-bearing structures. Now, intermediate filaments are much more stable than microtubules and microfilaments, and they're much less likely to be chemically or enzymatically broken down in cells, so they are significantly more stable. Now, as we wrap up this section and this chapter on the cytoskeleton, I don't want to leave you with the impression that the individual cytoskeletal elements work independently. Now, if you know anything about muscle structure, you know you've got actin filaments working with motor proteins, and you'd have seen microtubules with motor proteins moving along them. So you know that motor proteins and different parts of the cytoskeleton work together. But I want you to also appreciate that different parts of the cytoskeleton work together to give you the cell structure and the associated functions. So what we have is a situation where microtubules often resist bending when a cell is compressed. Microfilaments serve as kind of contractile structures which can generate tension, and then intermediate filaments are pretty elastic and tend to withstand tensile forces pretty well. So three kind of different structural characteristics and three different properties that these filaments bring to cells, but they're all working together, and so there's actually a lot of interaction between the intermediate filaments, the microtubules, and the microfilaments. So what we're looking at here is a protein called plectin, and as what plectin is doing is it's cross-linking the intermediate filaments here shown in green with these microtubules shown in blue. So here we've got one type of cytoskeletal element interacting with another type of cytoskeletal element. So at least appreciate as you walk away from this chapter that the cytoskeleton is highly regulated and the different elements interact with one another to produce the characteristics that cells need. So we'll wrap it up there. That's the end of cytoskeletal systems. I'll see you soon for our next chapter.